So hello everyone, I'm Jilan Ponsabalia and I am here with my group mates to report about molecular genetics, which is letter E, discussing the protein synthesis, post-translational modification, gene mutation, DNA repair, and transposition. And now, what is protein synthesis? So protein synthesis, a cornerstone of molecular genetics, occurs in two stages. So the first stage is transcription. Now, ang mahitabo sa transcription na ay tulog ka a process, which is first is the initiation, second is elongation, and third is termination. Now, kani tanan nga process is ang responsible nga enzyme ani is ang RNA polymerase. Now, in initiation, ang RNA polymerase binds to the promoter. In sa elongation, RNA polymerase makes a copy of the coding region using base pairing rules. And sa termination, RNA polymerase makes mRNA until it reaches the termination site where it stops. Now, sa makita nimo sa picture, diri sa nucleus, ang mahitabo diri sa nucleus ay ang transcription. Now, diri kaning makita ninyo na ay pre-mRNA. No, so mo na ang gibuhat sa RNA polymerase iyang gi-copy kanang red diha, mahimo siya og mRNA mo din ang mogawas sa nucleus and mo-travel siya sa cytoplasm and moadto sa ribosomes. Now, in ribosomes, diri din mahitabo ang translation. Now, in ribosomes, mosulod din diha ang tRNA nga mo-attach sa codon sa mRNA. Now, kaning tRNA or transfer RNA na siya anti-codon kaning tulo ka codon and gaber sad ni ang tRNA o amino acids. Now, ang tRNA mo ni siya mo-attach no sa masulod siya sa ribosomes, mo-attach din siya sa mRNA. Now, unsaon man sa pagkabalo sa tRNA kung asa siya mo-attach, mao nang importante kayo ang mRNA kay siya ang direct kung asa mo-attach ang tRNA no kung asa siya fit. No, sa pag-attach sa isa ka tRNA, no, magdala siya ang isa ka amino acids, no. Ang sunod din nga mo-attach na pud nga tRNA, magdala gyapon siya amino acids and magbuhat din siya og peptide bond mabilin to ang amino acids. And mao na nga ang process hantod sa moabot sa code nga UGA or UAG, no, which is stop nga code. And katuding mga amino acids nagbuhat og polypeptide bond, mao na din to siya ang protein. So pagkahuman sa protein synthesis na din mahitabo nga post-translational modification. Now, the post-translational modification beyond the linear sequence of amino acids, post-translational modification adds complexity to proteins. These alterations, such as phosphorylation or glycosylation, fine-tune protein structure and function. This dynamic regulation is crucial for cellular signaling, ensuring precise responses to internal and external stimuli. Now, there are many types of modification, but kanirang lima ako ang i-discuss sa inyo. The first is acetylation, which is adding acetyl groups to amino acids. This modulates protein structure and gene expression. And the second one is phosphorylation, adding phosphate groups to proteins, and this influencing protein folding, stability, and function. And the third one is ubiquitination. Now, this ubiquitination is attachment of ubiquitin molecules to protein. This marks proteins for degradation by proteasomes. And the fourth one is methylation, which is adding of methyl groups to amino acids. This affects protein-to-protein interactions and gene expression. And the last one is glycosylation. Now, this glycosylation is adding of sugar molecules to proteins, influencing protein folding, stability, and function.
So, hi everyone. Ako si Vladimir Scorpion. Karon akong i-discuss, i-report sa inyo ang gene mutation classifications. Sa gene mutations, natay patara ang atong makita ang point mutations, frame shift mutations, chromosome mutations, silent mutations. So, ito sa point mutation. Sa point mutation natay substitution, insertion, deletion. So, substitution, one nucleotide is replaced by another. Other term is can also refer to a replacement of one amino acid in a protein with a different amino acid. So, ina-replace niya, no? Kaya siguro kailangan siya i-replace kay para mas stable ang structure sa pag-mutate. And the next is insertion. Insertion, an extra nucleotide is added to the sequence. So, yan na siya para tungagan niya ang structure, no? Insertion para ma-achieve ang therapeutic effect such as conferring novel functions against different diseases or the protection against repeat infection. So, importante kining insertion kay siyang mag-protect para sa mga lain-lain nga disease o sa balik-balik ng mga infections nga ilang matagbuan. So, mao na siya ang insertion. Ang deletion, nucleotide is removed from the sequence. So, from the name itself lang, no, ginawala na siguro kay na yung mga defect or unsa ba mong na-deletion siya, lapin siya sa deletion. Our next is frame shift mutation. Sa frame shift mutation, nalamang tay insertion or deletion. Alter the reading frame, affecting all subsequent amino acid. So, ay mag-insertion or deletion ay mawala. And next is ang chromosomal mutation. Sa chromosomal mutation, natay upat. Duplication, deletion, inversion, o translocation. Sa chromosomal mutation, ang duplication is a segment of the chromosome is duplicated. So, dari na, no, makita na ito i-duplicate na niya, i-transfer niya into its homologous chromosome, giving it a duplicate of some genes. So, dari na nato makita ang pag-duplicate sa mga genes. And next is deletion. Deletion na po, segment of a chromosome is lost. Again, wala siguro sa mga factors. Bago siyang ma-deletion. Inversion. Ang inversion is a chromosomal segment is reversed. So, ma-reverse siya, no? So, reverse ang iyang yung structure. So, why ma-reverse? And next is ang translocation. Chromosomal segment moves to another non-homologous chromosomes. So, translocation. A segment from chromosome is transferred to another. So, may segment sa structure like example A, B, C, D, E, F, G, H. A padulong F na translocate siya sa PICAS. Ingana, no? Into another. Para na siya magbuong mutation. And then sa chromosomal mutation, dapat na to ang sa chromosomal mutation, dapat na to makita ang condition nga Down syndrome. So, next is ang silent mutation. So, kani siya, tahimik lang yun siya, silent. Nucleotide change, but the amino acid sequence remain unaffected. So, isang pag-down siya hindi mo, no? Isang pag-down niya mag-change. Pero yung amino acid sequence remain unaffected. Digid siya matandog kay silent mutation. Good. And thank you, Marata.
3 types of transposable. First is DNA transposons. Ang DNA transposons, i-direct niya o cut and paste ang mechanism padulong sa DNA sequences. Second is DNA heterotransposons. Long terminal repeat heterotransposons contain LTRs at both ends. Heterovirus-like replication mechanism. Non-LTR heterotransposons lack LTRs, use an RNA intermediate during transposition. Last is autonomous and non-autonomous elements. Ang autonomous ay encode niya ang proteins necessary for transpositions. Ang non-autonomous ay depend na siya sa proteins na gi-encode sa autonomous.
DNA repair system. Here are some simple explanation and example of each DNA repair system. Number 1. Base excision repair. BER. Correct small non-helix distorting base lesions such as damaged or incorrect bases. Example, repair of the single damaged base like uracil by removing the base and replacing it with the correct one. Now let's talk about the nucleotide excision repair. NER deals with larger lesions that distort the DNA helix such as UV-induced timing tumors. Example, repairing timing tumors caused by UV radiation where a segment of damaged DNA is excised and replaced. Mismatch repair. MMR corrects errors that occur during DNA replication, fixing mismatch base pairs. Example, correcting a mismatch pair like AC instead of AT in the newly synthesized DNA strand. Double strand break repair. Homologous recombination. HR uses a homologous DNA sequence as a template to repair double strand break. Example, repair a double strand break by using sister chromatid as a template. On the other hand, non-homologous end joining directly rejoins broken DNA ends without requiring a template. Example, ligating the broken ends of a DNA double strand break without using a homologous template. Lastly, we have a direct reversal repair. This repair system directly reverses specific DNA lesion without removing or replacing nucleotides. Example, repairing DNA damage caused by alkylating agents where the alkyl group is directly removed from the DNA base.